It is great to be here. Very nice to see so many people here this morning. And I'm also from the University of Copenhagen, so I think it's quite remarkable to be here in this ceremonial hall. My office is a little less spectacular, but it's good to be here. So I think I speak for many of us when I say that we would really like to live until we are 100 years old. And I would even like to live like a good, healthy, active life until I'm 100 years old. I think many of us want to do that, and I think that's actually a goal that is within reach. And to obtain that goal, we really need to be able to better maintain and preserve our skeletal muscle mass and function. Unfortunately, we know that insulin sensitivity, the ability to regulate muscle hypertrophy, so muscle atrophy, occurs with aging. And also there is a marked decline in mitochondrial function, so the organelles that produces the energy we need to move. And this is impaired in aging, and this impairment is further accelerated if we get cancer or if we get other aging-related diseases. So, fortunately, there are an old trick by which we can circumvent these problems. And this is exercise. We all know this. We've heard about this so much. So if all of us could just obtain an optimal amount of exercise, many of the challenges and the age-related problems that we are all here now today to learn about and fix would actually not be an issue. So why is this old trick not working in populations? And that is, of course, because we do not exercise enough. So we need to find new targets, and I think that if we could look towards some of the targets that are regulated by exercise, specifically in the skeletal muscle, this might give us some inspiration as to how to try to at least get some of the benefits of exercise. And we have just been through one epidemic of COVID, of course, but we still have at least one other epidemic that we're facing, and that is the epidemic of obesity. We have around 1 billion people being obese, and this obesity epidemic, it plays out as a private struggle, but it's also a major public health crisis because we know that obesity increases the risk of cardiovascular diseases. It also increases the risk of at least 14 different cancers, and it overall impairs healthy aging. And I've called it the sarcopenic obesity epidemic here, and that is because in obesity, there is very often a marked decline in skeletal muscle mass and function. So even though the body mass is large, the relative amount of muscle mass is very much in decline. And this really impairs the ability to perform everyday tasks, but it also contributes to the impairment in metabolic control seen in obese people. And this is actually has quite a devastating result. This sarcopenic obesity, so this decline in muscle mass, increases the odds ratio for different comorbidities that are already associated with obesity. It also quite markedly increases the odds ratio for different metabolic disorders, including diabetes and dyslipidemia. It also increases all-cause mortality, which I think is also, of course, a very hard endpoint. And also, I think, particularly very important for the elderly population, that this sarcopenic obesity also has really marked effects on prolonging the length of hospital stays, and it also markedly increases post-operative complications for the patients. And most elderly people, they do go to hospital, and they do have already hospital stays. So when you then have sarcopenic obesity, these are quite markedly prolonged. So I think you get the picture that this is also very clinically important. But as Jens talked about earlier, there is, I think, a very fantastic and good solution in sight with these new GLP-1 based drugs. That really works fantastic, and they are, of course, accessible to the wealthy parts of the world. And these data I'm showing here is just from the STEP 1 study, where they show this once-weekly semaglutide usage in people who are overweight or have obesity. And here you can see this really remarkable decline in body weight. So the issue with these otherwise fantastic data that Jens talked about earlier, they are fantastic, full stop. But everything good can be improved. And this is that together with this weight loss, there's actually quite a marked reduction in muscle mass as well. And this has been shown now in many different trials in diabetic and obese populations, showing that for a weight loss of around 16 kilograms, around 7 kilograms of this is skeletal muscle mass. So if you just show it in a different way, it's about 40% of the body weight that is lost that is skeletal muscle mass. So if we could avoid this muscle mass loss, this would likely be beneficial, in particular for an elderly population. And we know that muscle mass is extremely important for our survival. This has been shown in many different prospective studies, and I bring this one, which is an old one, but I think this is one of the very high quality studies really showing the importance of muscle mass and strength in survival in the elderly population. If you just focus your attention on these bars to your right, this is in people above 60 years of age. And then it just shows all-cause mortality in the weakest third of the population compared to the strongest third of the population. And I'm sure you can appreciate that survival is twice as high, or mortality is twice as low in the strongest third. So I think these data are really remarkable, and probably also why there is now really this rush to better preserve muscle mass also in the industry. And this is just one review I put up where they have focused on this and where the different trials are, but there are many more because this is also a concern in the cancer cachexia field. So there are also a lot of companies working to try to restore muscle mass. But it's of course also important in the obesity space. And then there is Versanis that was last year bought by Eli Lilly, who produces bimagrumab that Eli Lilly now tries to combine with the tirzepatide drug to prevent this muscle mass loss. So the skeletal muscle really takes a hit in obesity and during weight loss. So to reduce the impact on the muscle, one could really cultivate the thought of pairing these obesity drugs with some drugs that mimic exercise or activate exercise-induced signaling pathways. And I think the benefit of that is illustrated in this study, this is a slide study that was conducted here in Copenhagen, where they combined liraglutide, so the precursor of semaglutide, with exercise. And what they showed was, first of all, that the weight loss was more effective. But what they also showed was that the weight loss was obtained with a much less reduction in muscle mass, so with a healthier weight loss. They also showed that if they added exercise, the weight gain that occurred after the intervention period was much less. And they also showed that the weight that was, so usually when the weight is gained after these drugs, about 80% of the weight that is regained is actually adipose tissue, and this was also diminished in the people who had exercised. So there is something within the skeletal muscle that is really worth pursuing to combat obesity. And we have worked on this for quite a while, taking different approaches, and I'll just focus on one target that we are interested in today. Because we know that exercise is an old trick. So what we have done is that we have mined all of these emerging proteomics, phosphoproteomics, also metabolomics studies that are spitting out now, and we are now trying to understand what are these exercise signatures, what are the druggable targets that are induced by exercise. So with this mindset, we've really tried to mine these different proteomic data sets, searching for interesting candidates. And I just want to tell you one little thing we noted is that with all of these proteins being regulated by exercise in the skeletal muscle, about only 10% of them, we actually know their mechanism. We know what they're doing. The remaining 90% of them has unknown mechanisms. We have no idea what they're doing in this muscle. So these omic tools, they're really fantastic for discovery, but we still lack these functional annotations to the players identified. And this is what we have tried to do. And we have the selection pipeline that we're working on. And I just want to show you one little vignette of one of the proteins that we have looked at. And that is a protein called TNIC. So TNIC is a kinase that is very heavily expressed in the neurons in the brain, but it's also heavily expressed in the skeletal and the heart muscle. It has been associated with JNK signaling, NF-kappa beta signaling, Wnt signaling, and also AMPK signaling. And our attention was spiked mostly by its induction by AMPK, because AMPK is, first of all, it's a target of metformin, but it's also one of the proteins that heavily activated during exercise in the muscle. So we did an initial study in Drosophila, and this was a screen we did together with a group in Helsinki, where we noticed that the fly ortholog of TNIC, called misshapen, if you knock it out in the fly, they become completely metabolically inflexible. And that means that normally our cells, they need to be able to shift between substrates they use. They can use fat, or they can use glucose, they can even use proteins. And this shift is essential for our survival, and it's also something that's a little bit, I think is emerging, that is impaired in aging. But these flies that lacked TNIC, they were completely unable to shift from a high protein to a high sucrose diet. They died. And we then did targeted metabolomics, and here I'm just showing some of the metabolites that were changed in the wild-type flies when we changed the diet, and they were not changed at all in these flies. So they're really showing this metabolic inflexibility, and we think that it has something to do with that they're actually completely incapable of turning on de novo lipogenesis, and that was indicated by a quite remarkable reduction in the gene levels of FAS and ACC that facilitate the synthesis of new fatty acids, and they were markedly reduced and could not be upregulated by diet, as in the wild-type flies. So now, knowing that exercise regulated TNIC in the muscle, also knowing that TNIC had these really remarkable effects on substrate shifting and lipid metabolism, we felt that this was worth pursuing. So we generated the knockout mice. And here I will give full credit to Fung, who is a postdoc in my lab, who has done all of this TNIC work. And she placed wild-type litamate mice or TNIC knockout mice either on a normal chow diet or a high-fat, high-sucrose diet, supplemented with glucose water to mimic the Western diet of us, what we eat. And we are normally blinded when we do these experiments, but that was impossible in this case, because this is a wild-type mouse on a high-fat, high-sucrose diet, and this is a knockout mouse on a high-fat, high-sucrose diet. So they were completely protected against this diet-induced weight gain that we normally see. And it was not because this mouse ate less than this mouse. They ate the same, or even more. If anything, the TNIC knockout mice ate more. So this is my dream phenotype. I would like to be able to eat as much as I can, stay lean. So I would very much like to be this mouse. And what we noted was also that it was an incapability to gain fat mass that these mice exhibited, because they had completely preserved lean body mass, but they just wouldn't gain fat. And that was despite they ate all of this fat. So we thought this was quite remarkable. And here I'm just showing the data, so you can trust me on that they did eat the same, or if anything, they ate more than the wild-type mice. So of course, when a mouse eats the same, but they weigh less, we are looking towards something with energy expenditure. And this was also what we saw here, that we saw that in the dark cycle, which is when the mouse is awake and walking around, they did have an increased utilization of oxygen, indicative of increased energy expenditure. I will say that they also had hyperlocomotor activity, so they actually moved around more. And this is probably something to do with TNIC's effects in the brain. So then we, of course, wanted to see if these mice were also metabolically healthy, and in just the chow-fed mice, where we also did not note any differences in body mass, they seem to have similar glucose tolerance as the normal mouse. However, when we challenged the mice with this high-fat, high-sucrose diet, the wild types, as expected, became glucose intolerant. This is what we also see in humans, that we cannot clear off the glucose from our blood when we eat a glucose load. But this effect was completely prevented in the TNIC knockout mice. mice. So they were fully capable of clearing off their glucose despite this high-fat, high-sucrose diet. And they also had a general down-regulation of the insulin levels, really indicative of lowered or increased insulin sensitivity and better metabolic control. So we then looked a little bit deeper into the tissues of these mice, because insulin was introduced, so that was good. But insulin is really the hormone that controls our blood glucose. And insulin activates this quite complex, it's very simple here, but quite complex and highly controlled intracellular signaling cascade that results in the translocation of these glucose transporters called GLUT4 to the plasma membrane. And this is required for the glucose to enter the muscle cells and the fat cells, because if it's not there, the water-soluble glucose cannot bypass the lipid bilayer of the membranes. So if the GLUT4 transporter is not there, the glucose cannot come in. And this is a vital step for regulating glucose uptake. So we measured the effects of insulin on glucose uptake using isotopic tracers in these mice. And if you just, because in the wild or in the control chow-fed mice, we didn't note a big difference. But in the wild-type mice that were placed on a high-fat, high-sucrose diet, they became insulin-resistant, as expected, but this did not occur in the TNIC knockout mice at all. They were completely protected from diet-induced skeletal muscle insulin resistance, and this was also the case when we looked at the wide adipose tissue. So they seem to be able to fully maintain functional insulin signaling. And another hallmarks of having a fatty diet and having obesity is that a lot of the fat is deposited in the liver. So normally when we place a mouse on a high-fat diet, the liver grows and it's packed with fat. And this is what we see in a wild-type mouse, but we actually did not see this so much, or at least to a smaller degree, in the TNIC knockout mice. So even though they ate all of this fat, they ate more fat than the wild types. They did not put it in the liver, and we're not exactly sure where they're putting it. But they did seem to have a little higher levels in the circulation, so maybe that's where. And we think it's because there was a downregulation of CD36, which is the protein that transports lipids into hepatocytes. And they also lacked upregulation of FAS, which was this lipogenic de novo lipogenesis gene. All right, so the last data is this one, because we felt that these data was quite exciting, but of course they had the limitation of being obtained in the fly or in the mouse model that is not us. And we're a little limited in doing this mechanistic study in humans, of course, but what we can do is that we can get data from these big databases, the GWAS databases. And here we looked at the Tattoo Knowledge Portal database, as well as UK Biobank, to explore any GWAS data sets that would associate any variants of TNIC with obesity or diabetes-related traits. And we did see that there was really a strong association between TNIC variants and random glucose, fasting glucose, HbA1c, and many of the other different parameters that we associate with dysregulated metabolism. So we thought this was exciting that we described this role. So in conclusion, I strongly believe that, and the data points this way too, that preserving muscle is really an unresolved challenge, and we need to solve it in an aging society. We know exercise works, and I think if we can figure out exactly how, we can maybe harness this knowledge therapeutically. And then I just want to take you over these different studies we did. We showed that TNIC knockout mice were protected against diet-induced obesity, and this was actually recently reviewed very nicely by Ewell and Alex here. And we have also now shown that this is likely also something that happens in the humans. And in parallel to our work, this study came out showing that TNIC inhibitors can be used in humans, that they are safe in humans. So I think that's really exciting to see if they can be combined with other drugs that maybe lower appetite, so that they two combined can maybe synergize. With that, I need to thank my research team and all of our funding and collaborators. And I want to thank you all very much for listening to me. Thank you.
Thank you so much, Lykke. That was really a fantastic talk. We have time for a few questions. Maybe I can start. So do you think that the TNIC effect is indirect? I mean, it's not through working directly in the adipose tissue? Do you think it's indirect, and how?
Yeah, how I don't know, but I think it is indirect. And the reason I think so is because we have also done cell studies where we have knocked out TNIC in the different cell tissues. And for example, the adipose tissue, TNIC is not expressed in the adipose tissue. We cannot find it there. So I think that's definitely indirect, yeah.
It's a fantastic presentation. I need to TNIC about it. The question I was waiting to get to, I'm sure many are asking here, first law of thermodynamics, we hear about longevity. What's the most consistent thing we've seen in prolonging mammalian life is calorie reduction. Now you're telling us we can calorie increase. So what was the length of life of these animals? Was there any compromise in that regard?
Yeah, we don't know because we didn't let these mice go until they would die by themselves. We put them into experiments before. But we did do some fly knockout studies. And they lived the same as the wild type. So we didn't see anything in the fly on longevity as such. The problem was if there was a change in substrates all of a sudden, then they would die. Then they couldn't handle that. But if they were just placed on the same diet and let go, then they would live the same, actually, yeah.
Thank you. Why did evolution put TNIC in us, and what is lost if we inhibit it?
Yeah. I think it's there because what our data points towards that it is required for changing substrates as the substrates are available. This is a very important mechanism because if you can only find meat one day or you can only find sugar the other day, you need to be able to change. So if that is true, I think that is a very important function of TNIC. Yeah. And your other question, what was that again?
What is lost if you inhibit?
Yeah. Well, this ability is lost. Yeah. And we did see, it's not all glitter, because we did see, for example, when we did an insulin tolerance test, some of the wild type mice, they just bumped back up in blood glucose. But some of the TNIC mice, they just kept dropping until they became hypoglycemic. So we had to stop the experiment. Yeah. So maybe that ability to sort of counterbalance is lost. Yeah.
Hi, Lykke. So very quick kind of intro. So the study that you published in Science Translation Medicine, Science Advances last year came just before last ARDD, and it has a massive influence on my life. So, like in every way, because we had a TNIC inhibitor in phase 1, and now we have it in phase 2. So it looks like it is also supported by methylation aging clocks. And I hope that today you will stay till like 6:20, where we're going to present this work. My question is that, do you think that, let's say, if there is TNIC inhibition or TNIC knockout, if you put those mice on additional exercise, if they will gain even more muscle?
Yeah, I think so, because I think they will run more.
By the way, this is TNIC inhibitor in phase 2. I made it Tiffany green.
Can I have it?
Humans take it already.
It's matching your shirt, Lykke.
Yeah.
Thank you. Could you comment on TNIC or TNIC inhibitor effects on brain function, or more specifically, brain energy metabolism?
I can't, because it's a little outside of my area of expertise, and I think I think I'm not the right one to answer that. And I think also with the inhibitors, the ones I've seen, I haven't seen any data that they would actually cross the blood-brain barrier. So I don't know this is a knockout mouse, so it has the knockout everywhere, but if you add an inhibitor, you might just get the effects on the periphery. But we do know that TNIC is expressed in the basal ganglia, which is heavily involved in movement. And we do see that these mice, they do move more voluntarily. So that could have an effect. But apart from the metabolism part, I'll just have to pass on that one. Yeah.
All right. Thank you so much, Lykke. That was really a fantastic talk.